Okay, let me share my screen. Okay. So I can't see the chat, so feel free to like interrupt me at all, or if you just want to type in the chat, I can answer it afterwards. So I'm going to be talking about alpha helices and beta strands. And again, all the information on these slides will be from either the textbook or the lecture notes, so you can always refer back to those if you have any questions after the session. So the most common location for an alpha helix to be found in a protein structure will be along the outside of the protein. So that's where you would like assume it would be. And that would mean that one side of the helix would be facing the solution, which would usually be hydrophilic, and one side would be facing the interior of the protein, which would be hydrophobic. So the specific amino acid side chains, which are kind of either inside or outside, tend to switch from hydrophobic to hydrophilic with a periodicity of 3 to 4 residues, because that's how long it takes to turn around the helix. Okay, and so here's kind of a drawing. The picture in A is structure of myoglobin, and you can see the helix in the middle is then amplified in the structure B. So you can see the yellow residues are hydrophobic and the red residues are hydrophilic, and you can see that the red residues are facing the solution, and then when you flip it over, the yellow residues would be facing the protein. So alpha helices that have one hydrophobic face and one hydrophilic face are known as amphipathic helices, and these are really common because of the nature of alpha helices being found on the edges of proteins. But that doesn't mean that you can assume that all alpha helices and globular proteins are amphipathic, because there are always exceptions. For instance, residues that are facing the solution can sometimes be hydrophobic. And alpha helices can be completely buried within the protein. They don't necessarily need to be on the edge, although that's rare. And they can also be completely exposed, but that's also rare. So it would most likely be that residues that face the solution can be hydrophobic. So when you look at these 3 helices, you can look at the lettering to see which amino acids they are, but are any of these amphipathic?
Is it the second one?
Yeah, so the middle one would be. So A is a completely buried helix, and so you can see that most of the residues are hydrophobic. And then C is more on the exterior, and so the residues are mostly hydrophilic. And then with B, you get a good example of an edge where there's half of the spiral is hydrophobic and half is hydrophilic. So when trying to determine if a helix is actually amphipathic, one of the easiest and most convenient ways to represent the amino acid sequence in a helix is through a helical wheel or a helical spiral. So each turn spans 3.6 residues. So each residue can be plotted every 360 divided by 3.6, or 100 degrees around the spiral. So if you're looking at this, you can see 1, 2, 3, 4, and each are separated by 100 degrees. And then this is as if you're looking down into the center of the helix. So residues on one face of the helix cluster together on one side of the spiral, and you can easily tell if a helix is amphipathic by just checking to see where the residues are and which ones are hydrophilic and which ones are hydrophobic. So beta sheets are a little bit more complicated. When beta sheets are found on the surfaces of proteins, they are usually amphipathic as well. But because of the nature of the beta strands, it's not as easy to necessarily discern from their amino acid sequence whether or not they're going to be amphipathic. In this protein, the strand has 3 buried hydrophobic residues, and they're arranged in the pattern of every other residue is hydrophobic. So you can see the 3 yellow ones on the interior. But like they're way more difficult to discern in sequence patterns than alpha helices, which you can kind of tell. Surface exposed side chains can always be hydrophobic. For instance, the valine here is hydrophobic, so you don't even see a pattern. And there are also fewer residues that span the protein domain because the beta strand is more stretched out. So you have less material to look at when trying to decide whether or not they're amphipathic. So now it's kind of a different topic, but still related. It's structural motifs. So simple combos of a few different secondary structure elements in specific arrangement have been found to occur frequently in protein structures. And when these combinations occur frequently, they're called structural motifs. And they're usually formed by packing side chains from alpha helices or beta strands close together. So these motifs can have a function, for example, binding to DNA, metal ions, or small molecules, but some are just part of larger structural and functional assemblies. So we're going to take a look at a few examples of motifs. So this is the first one, the helix turn helix motif. It's probably one of the more simple motifs that we would learn in this class. And it basically just consists of two alpha helices joined together by a short turn region, which would just be a few amino acids. And you can see from the drawing, it's pretty much exactly like what you would think from the name of the motif. And this motif will be commonly found in proteins that recognize DNA sequences because one of the helices can insert itself into the major groove of DNA, and the side chains on that helix will make sequence-specific contact with the bases that are inside of the groove. The second motif we're going to look at is the helix loop helix motif. It's very similar to the helix turn helix motif that we just talked about. The only difference is that there's a longer chain connecting the two helices, so with way more amino acids, and it's called a loop. So you can see the structural drawing again, and then it's called, another name for it is like the EF hand, because as you can see from this drawing, it sort of looks like the pointer, middle, and thumb. So this motif exists in calmodulin, which is a protein that responds to calcium levels in the cell. And the motif occurs 4 times, and it forms a calcium binding site, which you can see like it kind of fits into the hand right there. So beta strands also can be arranged in motifs, and anti-parallel beta strands can be connected by either turns or hairpins. And then one example of this would be 4 adjacent anti-parallel strands arranged in a pattern. It was just similar to the repeating unit of an ornamental pattern used in ancient Greece, and so it's called the Greek key motif, and you can kind of see it here. It's just the beta strands linked by amino acids. Yeah, not too complicated. This motif doesn't have a known specialized function, but it occurs frequently, so it's recognized as a motif. So parallel beta strands must be connected in a unique way because of their different structure. If the 2 adjacent beta strands are consecutive in the amino acid sequence, the ends must be joined, will be on opposite edges of the beta sheet. So you can't just use like a simple turn or a simple loop to connect them. The polypeptide chain actually has to cross the beta sheet from one edge to another. This is often done by a helix. And so the motif consists of a beta strand followed by a small loop, followed by an alpha helix, followed by another loop, and then the second beta strand. And so you can kind of see what I mean here. You couldn't just connect this by a loop because it wouldn't be connecting the correct ends. It would be connecting a C to C, and so that wouldn't work. So you need to have an alpha helix going down the middle and reconnecting it. So when forming these motifs and when alpha helices packed together, they can only assume a few orientations because of their geometry. So the most common way that alpha helices will be packed is by fitting the ridges formed by a row of residues separated in sequence by 4 in one helix into the same type of grooves in another helix. And basically these ridges and grooves will end up forming an angle of about 25 degrees to the helical axis. So if you're looking at these photos, it's like the helical axis is going straight up and down, and then the curvature will be coming like around from each one dot on one line to the dot on the next line. So basically, this is one helix and this is another. They're identical for the sake, and then you flip the second helix over 180 degrees and then rotate it 50 degrees, which allows it to pack into the helix. These are sometimes kind of hard to visualize. So I would advise like looking at the pictures in the book and also trying to draw it out for yourself to see how it works. They can also fit in another way, which is by fitting the ridges formed by amino acids 3 residues apart into the grooves of amino acids 4 residues apart, or vice versa. So the first ridge will form an angle that's 45 degrees to the helical axis, seen here, and the other type will make an angle 25 degrees in the other direction. So you can see how they're rotated. And then after a 190-degree flip of one of the helices, they'll be on the same side and they'll be able to line up with a 20-degree rotation. And then these structures kind of show you, you can use these to help visualize the 1, 2, 3, and 4 images by looking at these dots. Okay, that is it. Does anyone have any questions? Okay, if not, then I'll pass it on to the next tutor.
Sorry about that. Let me share this again. All right, can everyone see the PowerPoint? I'm just gonna take that CS. Okay. Hi everyone. So I'm gonna be talking more about alpha helices, specifically talking about some superhelical structures that can form, coiled coils. We'll be talking about heptad repeats, and again, we'll go over alpha helix packing briefly. So coiled coils, when you have a single alpha helix in a solution, it's not very stable, but it can stabilize itself by packing hydrophobic side chains against another alpha helix. That can form what we call a coiled coil, a picture shown down here. And just so you know, parallel forms are more common than the anti-parallel ones. But an important thing to note is you have 2 right-handed helices, right? 2 alpha helices, right-handed. Those come together to make one left-handed coil, which we also call the superhelical structure. Let's look at what makes this left-handed coil. So as Heather talked about, normally we have residues per turn as 3.6, right? So in every 2 turns, we'll end up having 7.2 residues, excuse me. And we can see that at this picture here. If this is the first residue, I, and we were to wrap our way around in a right-handed sense, after 2 turns, 7.2 would, 2 turns would be here, and that would be 7.2 residues. But of course, we can't have decimal amino acids. So the 7th residue, I plus 7, ends up slightly left to the center of this helical axis. So now take the same alpha helix, make another one here, a red one, but rotate it 180 degrees. If you were to put these 2 on top of each other, what would happen? These residues wouldn't line up. So this I residue, these would line up, of course, if you did that, but these I plus 7 residues, one of them would be to the left of the axis, the other one, as pictured here, would be to the right. These would not line up. So to do that, the alpha helix wraps around in a left-handed fashion such that these I plus 7 residues line up. So we see here the I ones line up, and then as it wraps around to the left, now this I plus 7 residue will line up with the I plus 7 of the blue helix. And important thing to note, 3.6 residues per turn, that decreases it to 3.5 residues per turn when it does this left-handed superhelix. And if we look at the math here, now 3.5 times 2, 7 residues in 2 turns. That lines up perfectly. And this is just another note, so you know 3.6 becomes 3.5. Now, let's look more into the types of interactions we can have. So this is the heptad repeat. It's the pattern of amino acids. It repeats every 7. We label them A, B, C, D, E, F, G. There's a good, more vertical picture in your book on page 155 where you can see them actually interacting. But for now, we'll deal with this image because you'll probably see this on tests in this fashion. So let's look at some of the positions. So these A and D positions, they're in the interior of this coiled coil. Those are almost always hydrophobic. That makes sense. We know about hydrophobic cores, right? Those like the cluster in the middle, away from the aqueous environment. One thing to note is that these A and D positions can also be polar sometimes, because with those polar residues, you have H bonds stabilizing. Another important thing to note are the E and G positions on the edges at the top. Those are usually charged residues, so your lysine, arginine, glutamate, those sort of things. And those form favorable salt bridges, so ionic attractions. So there is a difference, as you can tell, between the parallel coiled coil at the top and the anti-parallel. Note the differences when you study this. So A, in the anti-parallel, A interacts with D, and then G, G, E, E, parallel. It's different, A and A, D, D, and then G, E. And that's important to note because those ion pairs at the G and E positions can decide whether this coiled coil is parallel or anti-parallel. Let's take, for example, just this top part of the parallel. If this G position were a positively charged amino acid, and this one were negative, that would be a favorable interaction, right? Positive and negative, attractive, and an ionic attraction. So that would favor the parallel coiled coil. But if you were looking at the sequence and you found that this G position were positive and this E prime position were also positive, you'd have repulsion from those two. And thus, in that case, this parallel coiled coil wouldn't be favored. That's what the problem on the worksheet is about, so just keep that in mind when you answer it. Now let's talk about packing, which Heather went over. I'll just go through it quickly as well. So usually in proteins, we actually, rather than coiled coils, we may find this hydrophobic packing, which, and for example, maybe you have 4 alpha helixes that'll orient themselves such that their hydrophobic residues face inward. So you have a hydrophobic core and then 4 alpha helixes. There's a good picture in your book on page 157 if you want to look at that. But you'll take the ridges of one helix and put them into the grooves of another helix. And I'll just go through the distinction. So if we're looking at one alpha helix, such as right here or right here, there are 2 types of ridges and grooves you can have. There are ridges and grooves formed by amino acids 4 residues apart. That's termed I plus 4. And if you were to count, let's say from here, so I plus 1, I plus 2, I plus 3, I plus 4, and you did that all the way down and drew a line like they did here, you would make these I plus 4 ridges and grooves. That'll give you a 25-degree angle off the helical axis, which is just down the center, right? So remember that. So that's one type that the alpha helix can form. The other type would be for 3 residues apart, do the same thing. One, or let me move this around. You start here. Oh no. 1, 2, 3, 4. Same thing, you get an angle of negative 45 degrees, but in the other direction, so counterclockwise, that's why it's negative. So keep that in mind. A single alpha helix can form either this type of this type of ridge or this type of ridge. And then with those types of ridges and grooves, you have 2 types of packing. You can have I plus 4 ridges into the grooves of another I plus 4. That's what's pictured in A right here. So to do this, take look at the one that's going to be your ridges. So in this case, that's the blue one. Take that, pull it out of the page, flip it 180 degrees and paste it back in. That's what you get here. And then rotate it such that the ridges of the blue, these lines with the dots, go into the spaces or the grooves of the red helix. So that's what we mean by the ridges going into the grooves. So you'll rotate that. For I plus 4 into I plus 4, you get positive 50 degrees. For I plus 3 into the grooves of I plus 4, that's the other one. Do the same thing, rotate it 180, rotate and then twist it such that these line up. You'll end up getting negative 20 degrees. I don't know how important this will be. When I took the class, I don't remember we had many questions on it, but we'll go over it just so you know. But just a note, these resulting angles, even though the sequence may change over time, the protein sequence, we find that these angles stay relatively consistent when we measure them in proteins. That's an example of conservation, which I believe you'll be going over in a couple lectures or so. But that's all I had. I can go over the problem since I have a couple minutes, but does anyone have any questions about what I talked about?
I have a question.
Yeah.
So when you are talking about the packing, why do we have to flip it like 180 degrees?
So that's just an orientation thing. That's, I believe that's the only way they'll line up. And
Okay.
I think it also helps for visualization, though I'm not exactly sure about that.
Okay, it's fine. Thank you.
Yeah. Anyone else?
Hello, I have a question.
Uh-huh.
What kind of interaction is involved in this kind of packing? I mean, hydrophobic side chains interacting with each other?
Yeah, you do have that hydrophobic interaction. I believe another term for it is just hydrophobic packing. So yeah, and I'm sure there's also, H bonding involved in some cases, maybe some ionic interactions as well. But I would say for the most part, hydrophobic.
Thank you.
Mm-hmm. How much time do I have? Okay, in the last 2 minutes, I guess we can go over the problem, which is on your worksheet as well. But assuming we're at physiological pH, which is around 7.4, so note that your charged residues will have their positive or negative charges. You look at the heptad repeat sequences for 2 alpha helices, shown here, helix 1, helix 2. They're in the coiled coil right now. So based on what you know about heptad repeats, is this coil parallel or anti-parallel, and why? Anyone have any ideas? And I put the image here from my slides. This top one is parallel, bottom one is anti-parallel. Let's give it a couple more seconds before I show the answer. But if anyone has an idea, feel free to unmute yourself. Okay, that's fine. So let's go over the answer, which it'll end up being anti-parallel. And let's go over why. So let's first look at parallel, right? So if this left helix is helix 1, right one is helix 2, let's look at the E and G positions, these ionic interactions. So the G position here, and the first one is lysine, K, positively charged, and E is arginine, also positively charged. From what we talked about, that's not favorable, right? Because there will be a repulsive interaction. That's not favorable for forming the parallel form. But if we look at the anti-parallel form, it's G on G, E on E. So lysine with glutamate, positive with negative, favorable. Same thing here, aspartate with arginine, negative, positive, also favorable. So you get anti-parallel. And I think that's kind of a common question on tests as well. So that's why I put that on there. Are there any more questions about this? Okay, then the next tutor can go ahead and start sharing.
Okay, I'm going to be going over membrane proteins, which are proteins associated with the membrane. And the first thing we want to look at is the structure of the phospholipids. So here we're going to have a polar head group, and it's hydrophilic. It has a negative charge on the phosphate, positive charge on the nitrogen, and some ester groups. And here we're going to have some hydrocarbon tails. And these are non-polar because there's only CH bonds. Okay, and so when these phospholipids join into the membrane, it's going to make it very difficult for polar molecules to cross the membrane since the interior is all hydrophobic. So so they're not going to go in there, and that's because that if the polar molecules do try to get into the hydrophobic interior, they're going to lose the hydrogen bonding that they would normally have with water outside of the membrane. So it's going to be energetically unfavorable for the a polar molecule such as water to cross the membrane. And so this is a reason that there are a lot of membrane proteins by which you can exchange polar molecules, for example. So I'm going to go over some examples of membrane proteins. And here we have one alpha helix, and it traverses the plasma membrane once. And so we can call it transmembrane. Because it spans the entire plasma membrane. Here we have several transmembrane helices, so here we'll have 7. There's also these structures called beta barrels, which are made entirely of beta sheets. And something to note about these transmembrane proteins is that the amino acids in the hydrophobic interior are all going to be hydrophobic residues. Mostly. So we can group these proteins also into some categories. So integral membrane proteins are going to be permanently associated with the membrane. So the membrane is their home, they're not going to leave. And then, so these 3 here would be examples of integral membrane proteins. And you can also have peripheral membrane proteins, which interact with the surface of the lipid bilayer, but don't enter into the hydrophobic space. An example of that would be this protein here, with a fatty acid anchoring the protein. And so it's still going to be associated with the surface, but it's not going to enter the interior itself. Okay, and yeah, something else, that these alpha helices are connected by hydrophilic loops because these loops are exposed to the water outside the membrane. So these proteins have a lot of functions. Does anyone want to throw out some functions that they have?
They could act like a receptor. So like if a ligand comes in, it could act like a receptor and cause a signal transduction.
Mm-hmm. Yep. So receptors. And this would be like a G protein coupled receptor, which would look something like this. They can be for recognition, so markers for cellular identification, and then also transport. So you could picture some, like a water molecule going through here, which would be an aquaporin. Okay. So I've drawn another alpha helix here, and the bilayer is 35 angstroms in depth, and the so from that we can determine how many turns an alpha helix will have in here. So it's going to be 35 angstroms and 1.5 angstroms per residue, and 3.6 residues per turn. So you're going to get 6 to 7 turns, which is about 25 residues. Okay. Any questions on any of that? Okay. So, so the hydrophobicity of the lipid bilayer requires the formation of regular secondary structure within the membrane. When we looked at water-soluble proteins, we saw that protein folding was driven by the hydrophobic effect, with the formation of a hydrophobic core that reduces the surface area of nonpolar groups exposed to water. And this will increase the entropy of the water molecule, since rotation toward nonpolar atoms would lose hydrogen bonding. And we also know that secondary structure, the alpha helices and the beta sheets, satisfy the hydrogen bonding requirements of the backbone, since it lost those hydrogen bonds with water. And for membrane proteins, you still have this requirement for hydrogen bonding. And so it needs to form regular secondary structure in the membrane, and it needs to, so the peptide backbone needs to make hydrogen bonds with itself because the hydrophobic interior of the membrane has no hydrogen bond donors or acceptors. And there's a huge energy penalty to move a protein into the bilayer if the hydrogen bonding is not satisfied. So it is required to hydrogen bond with itself. So we can see also if you have an isolated alpha helix, it has, here it'll have internal hydrogen bonding, it's surrounded by water, and the water will compete for these hydrogen bonds. So it's actually favorable for this alpha helix that's isolated to unfold. So it's not stable on its own. Versus in a membrane protein, you have no water surrounding the alpha helix. And so if it unfolds, it's going to lose the hydrogen bonds. So the hydrogen bonds aren't going to easily break because the lipids don't form hydrogen bonds. Okay, so this one is stable on its own. And a note on beta sheets, so beta sheets, you're not going to find a single beta sheet in the membrane. It has to form a closed structure like a beta barrel so that all the way around, all the hydrogen bonding is internally satisfied. So all the beta strands are going to form hydrogen bonds with its neighbors. Are there any questions on that? Okay.
For the beta sheet, isn't it a single polypeptide? So, or is it made up of several polypeptides?
It can be one continuous. So you can have, so when you have the sheet, they need to be, or sorry, so when you have the sheets, they need to be like the amino acids need to hydrogen bond with each other so that you don't have any exposed surfaces like flapping out, because otherwise you're not satisfying the hydrogen bonding. So it could be one very long protein. It just needs to be a closed structure.
Okay, so it could be both as long as it hydrogen bonds between the strands, or of like the beta sheets, as long as it hydrogen bonds.
Mm-hmm.
It
Yeah.
it could be made up of one protein or more.
Yes, I think.
Okay.
I'm not entirely sure, but as long as it's closed, it should be pretty good for the membrane. Okay.
I have one more question, if you don't mind.
Oh yeah, sure.
In regards to beta sheets, is there like a specific form that's more favorable, like anti-parallel or parallel?
No, I don't think so. It really depends on the amino acid sequence and how those are aligning.
Okay, so but within the membrane, when it forms that barrel, it doesn't necessarily matter if it's anti-parallel or parallel.
Yeah, it could be both.
Okay.
Or either, I mean, yeah.
Okay, thank you.
Mm-hmm. So the hydrophobicity of amino acid residues is critical for membrane spanning regions, and the interactions between transmembrane helices within the same protein rely mainly on hydrophobic residues. You're not going to see that many polar residues because hydrogen bonding requires specific geometries, but if you do, they're going to be interacting in partners. So here I've drawn two alpha helices from the same protein interacting. You're going to have these hydrophobic residues. You can also have these hydrogen bonding partners. So here an aspartate and a lysine. But the reason that you don't see that many of these, and it's mostly hydrophobic residues, is that a mutation in a polar residue is more likely to be deleterious. So if you lose one hydrogen bonding partner, you're going to disturb folding, or like let's say you change this one to a lysine 2, it would be electrostatic repulsion, and then your protein is not going to fold as well. And a point mutation in codons for hydrophobic residues often give another hydrophobic residue. So the protein will still fold in much the same way, and so they're going to be less sensitive to mutation. But these polar residues are critical for protein function a lot of the time, like you've seen in bacterial rhodopsin. So yeah, they're still there. So integral membrane proteins are stabilized by van der Waals and hydrogen bonding. And so van der Waals because you have a lot of hydrophobic residues, and the hydrophobic effect isn't going to be the dominant effect for membrane protein folding, because the hydrophobic side chains can interact with the lipid tails or other hydrophobic side chains. And so let me write that down. And for hydrogen bonds, these are often important for the protein to adopt its correct structure. So if you have an incorrect hydrogen bonding pair, it's going to be very high energy. So for example, these residues will align, and then it'll give the protein the correct orientation within the membrane. And in contrast, the hydrogen bonds in water-soluble proteins are weakened due to competition with water, so these ones are especially stable. And if there are any more questions, I want to quickly move to the worksheet problem, if that's fine. Okay, so the worksheet problem has two Ramachandran diagrams, and I wanted to, so determine if these can be membrane proteins. So here we have one with all beta strands. And do you all think that this could be a membrane protein?
Yes, it can be an alpha helix.
So this one would be a beta barrel more likely, because it has all beta strands. And I'm running out of time, but I just want to say this point. So if you have this, which would have beta strands and alpha helices, so it would be more of a random coil, you need your membrane proteins to be either all beta strand or all helical. And if you have a random coil structure inserted into the membrane, the backbone hydrogen bonds don't form, and this is going to be a big energy penalty. Okay, and so I think I'm out of time, but so the next tutor can go.
Hi everyone, can you see my screen?
Yes.
Okay, thanks. So I'll be talking a little bit more about membrane proteins, and I'll be going over some specific examples. I saw some questions in the chat about hydrogen bonding and beta sheets. So I can go a little bit more into that in my presentation. But first, I just kind of wanted to go a little bit, drive home the point about the hydrophobic effect and how hydrophobic molecules react in aqueous solution and how that forms certain structures in the cell, right? So you guys talked about proteins and proteins forming in aqueous solution through the hydrophobic effect. And the logic there is really similar to the self-assembly of lipids. So if you think about it, lipids have a hydrophilic head and a hydrophobic tail, which Ryan talked about, right? So if you put lipids in solution, they can self-aggregate to form these structures. They don't need anything extra. There's something intrinsically, there's an intrinsic property that allows them to form these ordered structures. So what is that? So why do lipids self-assemble? I think a lot of us are told in earlier classes that like dissolve likes, right? So we think that maybe the interaction between the tails is more favorable than interaction between the hydrophobic tails and the aqueous solution. That's not correct. The reason why proteins fold, the reason why lipids self-assemble is because by sequestering the hydrophobic parts of the molecules away from the water, you allow water molecules to engage in more hydrogen bonding with themselves. So people throw around the word energetic penalty, right? That's kind of like just like a shorthand that, but what does energetic penalty really mean? So if you imagine this bilayer, right? And then outside you have your aqueous solution. If I take a water molecule from the solution and I stick it inside the membrane, the energetic penalty comes from the fact that you lose the stabilizing hydrogen bonding from the outside and you do not regain it when you stick it inside the membrane. The hydrogen bonding, the dipole-induced dipole interaction of the water molecule and the tails is actually stronger than the tails between themselves, right? So the energetic penalty comes from the energy difference between hydrogen bonding in the environment and the loss of that stabilizing bonding when you stick it inside the lipid membrane. So how does this relate to protein folding in lipid bilayers? So as Ryan talked about, we have some alpha helices and beta barrels can form structures that are stable in these lipid bilayers, right? So, but the driving force of protein folding in lipid bilayers isn't the hydrophobic effect, right? Because you're not running away from water. You're inside this hydrophobic region with these fatty acid tails. So why is it that this is stable? And it's stable because when you take alpha helices and you put it in solution, the hydrogen bonding in the backbone competes with hydrogen bonding between the backbone and the aqueous solution, right? So regardless of whether you have hydrogen bonding between the molecule itself or hydrogen bonding between the backbone and the aqueous solution, either way, you still get your hydrogen bonding. But when you're inside the lipid bilayer, you can't get hydrogen bonding with the aqueous solution because you're in the lipid bilayer. You can only satisfy your hydrogen bonding by hydrogen bonding to yourself. So that's why alpha helices are generally stable. Isolated alpha helices are generally stable in lipid bilayers, but are not generally stable when isolated in aqueous solution. And going back to the beta barrel and hydrogen bonding, which I think someone mentioned in the chat, the reason why we need beta barrels to form in the lipid bilayer is that, so if you imagine they're flat, right? You have intramolecular hydrogen bonding between the sheets inside, but at the edges, your backbone is still exposed. So if you take a beta sheet from solution and you stick it inside the lipid bilayer, you don't regain the hydrogen bonding at the edges. So in order to regain that and reduce the energetic penalty, you have the sheet wrap around itself so that the hydrogen bonding is satisfied internally. So that's why we have beta barrels and hydrogen bonding inside the lipid bilayer. Does that make sense to everyone? Does anyone have any questions about this?
I have a quick clarifying question.
Yeah.
So So I understand the idea that you can't have beta sheets in the membrane because then you're not gaining back those hydrogen bonds that it originally had when it was in like an aqueous solution. But when we go back to the Ramachandran diagram, when I think Ryan had that diagram where it was like predominantly in that beta region, if we were shown something like that, is it okay to assume that those beta regions would form a beta barrel, or if it's in the beta region, it's just a beta sheet? Like, are we supposed to interpret one or the other?
I don't really remember exactly what diagram you're referring to, but I would say you can't, of course there are always exceptions, right? It's not always going to be that if you have something on the Ramachandran diagram that indicates that you have a beta pleated sheet, it's always going to be in a beta barrel conformation. Like the beta barrel is a unique example of the beta pleated sheet. There are a lot more examples of unique beta pleated sheets, right? It's not always, oh, if you're a beta pleated sheet and you're in the membrane, it must be a barrel. So I would say generally you can conclude that it's a beta pleated sheet. You cannot conclude that it's a beta barrel.
Oh, I see. Okay, thank you.
Yeah, sure. Does anyone else have any questions?
So it becomes a beta barrel because it doesn't want to form hydrogen bonds again or something?
Because it does want to form hydrogen bonds again. So if you look at, can you guys see my cursor?
Yes.
Okay, so if you look at this, right, you see the hydrogen bonding, the molecule hydrogen bonds to itself, right? So these two strips, it's the same molecule, it's one polypeptide. But at the edges, you see this carbonyl oxygen, and then you see the amine groups right here. There, if you didn't wrap them around, they would be interacting with in order to get the hydrogen bonding lost by moving the beta pleated sheet into the bilayer, the beta barrel allows the molecule to wrap around itself to satisfy that hydrogen bonding requirement.
Hey, Nat, I think we...
Sorry, everyone. I think my internet kind of collapsed, but I will finish up real quick. Sorry, everyone. Can everyone see this okay?
Yeah.
Okay, I will just go through this really quick. So here, the point is that you can just transfer ions through the membrane by having these aspartate residues to carry the proton through a wire of water molecules. So the water molecules aren't actually moving, which just allow the transfer of the proton through the channel so that you don't have any of this energetic penalty when dealing with the lipid bilayer. But when you do that, the proton can move in either direction. So if you stuck this into a cell membrane, let's say your mitochondria, your bacteria, the gradient of protons would just dissipate and your cell would die. So how do you control directionality? So bacteria have developed this protein called bacteriorhodopsin, where you have this retinal chromophore that allows the movement of these amino acid residues that transfer the proton in a specific direction. So in bacteriorhodopsin, here in the first step, you see that retinal is in all trans conformations. You look at these double bonds, they're all trans. And it forms a shift base with a lysine residue at position 216. A shift base is just a fancy word for a nitrogen double bonded to a carbon and covalently bonded to something else. So this shift base is protonated. So when light hits the retinal, the retinal can absorb it because of the conjugated pi system. So there's an energy gap consistent with the wavelength of light. It allows for the retinal to change conformation so that you get this cis bond at carbon 13. So the cis bond allows it to move downwards and protonate the aspartate residue at position 85. So once it protonates at the aspartate at position 85, the protein, which is just like a collection of transmembrane alpha helices, undergoes some sort of conformation that allows the shift base to move upwards, right? And this in part occurs because the shift base is no longer charged, it's neutral now. So once it comes up, it can pick up another proton from the aspartate residue up here. And this becomes protonated, and then you start your cycle all over again. I know I went through that really quickly because I think I'm running out of time, but does anyone have any questions about that? If not, I will stop sharing. Sorry about the Wi-Fi issues, everyone.
Okay, so if no one has any more questions, I'm just going to go ahead and stop the recording, and we'll hand it over to the tutors from the problem set session in a few minutes.